of 42 or more drugs which are available. Then premature mortality, depression, cognitive impairment, anxiety, these are the comorbidities which are there even if the patients are treated with these drugs. And SODAP is, sudden unexpected death in epilepsy patient is another issue. So what we need to have a good epilepsy research in India, but is it that we are not, there's no research in epilepsy? There's a lot of research going on in epilepsy. If you see over the last 3 decades, there have been spectacular advances in all fields of epilepsy, from the basic research to clinical diagnosis, investigative procedures, epidemiology, etiology, genetics, and issue of its comorbidities, psychosocial implications, as well as therapeutic options for the treatment of epilepsy. There has been a tremendous increase in the number of journals, books, and other publications available, as well as the websites which are dedicated particularly to epilepsy in this duration. There are numerous and well attended local and international meetings all over the globe covering specific, more general aspects of epilepsy. If you see, there is Epilepsy Society, there is International League Against Epilepsy, and there are well dedicated societies which are meeting frequently to discuss how to go further in the epilepsy research. However, in the epilepsies, like other medical diseases, there are well clarified areas of agreement and knowns, but there are also significant and sometimes diverse opinions and debates on important and still developing areas of semi-known or unknown areas. So these are the areas where we should ponder upon and work in tandem. So epilepsy research benchmarks is one of the guiding document, globally guiding document, that where the research in epilepsy is going on. And if any aspirant, new student, new researcher want to do research, this will be a guiding document for him that what should he do in the epilepsy research. So epilepsy research benchmarks are you can say available on the NINDS website, National Institute of Neurodegenerative Syndromes, Diseases. Uh, this is epilepsy research benchmarks are shared by the community vision for the research needed to expand our understanding of epilepsy and how it is treated, contributing to meaningful advances in health outcomes for the people with epilepsy. The epilepsy research community uses the benchmarks as a framework to focus research and benchmark progress over 5 to 7 years. So latest one which are available are 2020 epilepsy research benchmarks. These benchmarks are divided into further 4 sub areas. So we can choose which area we would like to have interest more and which is more socially relevant to the country where we are residing and doing research. So first area is to understand the causes of epilepsies and their relationships to epilepsy associated neurological, psychiatric, and somatic conditions. In this, what is happening? Identification of genes and pathways which are associated with all the epilepsies and epilepsy related conditions, and determine how changes in genes alone or in combination with other factors contribute to the development of these conditions. Then next is identify and understand the mechanisms by which infections, immune modulation, age, environment, vascular changes, perinatal effects, trauma, and other things alone or in combination contribute to the development of epilepsies and epilepsy related conditions. Then determine how alteration in molecular and cellular function interact with the alteration in circuit and network function in the pathogenesis of cortical hyperexcitability and in the clinical epilepsies. Identify and understand the mechanism by which factors related to age, gender, race, ethnicity, socioeconomic status, and the other features of specific populations affect the risk of developing epilepsy and epilepsy related conditions. So, in a nutshell, if you see area one is understanding the causes of epilepsy, maybe genomic, maybe infectious, maybe ethnic reasons, maybe age related, maybe economic and social. And you can say the relationship between these mechanisms that underlie the epilepsy is to be understood as far as the mandate of area one is there. So this is a very interesting area. If somebody want to use, you can say having interest in finding how epilepsy is induced, and still it is unknown. That's one more thing. There are some hypotheses which have been proven, but there are a number of things which are still unanswered. So one can work in this area, and this is a very interesting area for basic pharmacology studies. And second is area two, that understands how to prevent the epilepsy and its progression. If we are very good in area one understanding, or if we get good information from area one researchers, then this is very easy. This will be rather easy, not very easy, I'll say. So here we are understanding epileptogenic processes involved in epilepsies with neurodevelopmental reasons, including those due to genetic or epigenetic causes. Uh, there's one notion that most of the time epilepsy is genetic, okay, but it is not only genetic, it can be acquired also with time. So there can be genetic as well as epigenetic causes for this. Then understand epileptogenetic process involved in the development of epilepsy following traumatic brain injury, stroke, brain tumor, infections, neurodegeneration, and other insults to the brain. So this looks to be similar to the area one, but here we are talking about specifically inducing the disease through these things and then finding what are the molecular mechanisms they are affected by this. Then these are sort of interventional, you can say, area of that. That was the observational area, first one. So identify biomarkers that will aid in identifying, predicting, and monitoring epileptogenesis and disease progression, including biomarkers early after injury, insult, and identify those people at risk for epilepsy. So in every disease, now what happens, it is a current trend that we should know how early we can diagnose, how the better is that. So this thing is happening in all the type of chronic diseases. So if we can see the chronicity of a person towards a particular disease, then we can make a preventive measure to that. Develop and refine models aligned with the etiology of human epilepsies to enable improved understanding of epileptogenesis and rigorous preclinical therapy development for epilepsy prevention or disease modification. This is another area where the basic pharmacology studies can be carried out, where you can develop animal models which are having very good phase validity, construct validity, or predictive validity. So if all the three validities are there in the model, that would be excellent. So identify new targets and develop interventions to prevent or modify epileptogenesis and the progression of epilepsy and epilepsy-related conditions. So there is a problem that there are Me Too type of drugs. If you see, most of the drugs which are discovered, they are either copied as a template from the old chemical molecule, or they are having similar mechanism which is earlier, you can say, explored by the other drugs. So these drugs, we generally say Me Too type of drugs. They are not the new, you can say, drugs. They are just copy to the available drugs, and they will share the same pharmacological profile almost and the same side effect profile. So that will not be adding much to, we can say, the management of epilepsy. The combined complex system or machine learning approaches with laboratory studies in order to identify convergent phenotypes or pathways, examine background genetic or epigenetic effects, and consider novel molecular reclassifications of disease and epileptogenic process. As ma'am, Vice Chancellor ma'am was telling today in the inauguration, when I was attending that inauguration, she was talking about the use of AI, machine learning, and other in silico tools in current research era. So this is the same thing. We can, because we are having number of articles, a lot of genomic and proteomic information that can be processed or churned to get some new information useful to us for management of diseases. Area 3. In the area 3 of the benchmark, we have to search for the improvement of improved treatment options for controlling seizures and epilepsy related conditions while limiting side effects. As I told you, one of the limitation of the current drugs is they are not able to take care of comorbidities. Okay, that is one area where we can talk about. And then we should understand the mechanisms of initiation, propagation, and termination of seizures at cellular and network level for different seizure types so that we can devise the right therapies for these type of seizures. And coming to another big issue, that is refractory epilepsy. 30% of the patients which are refractory to the existing drugs or resistant to the existing AED drugs, they are supposed to give some better, you can say, pharmacological treatment, because they will be either dependent on surgical interventions or neurostimulation procedures. So mechanism, though they have their own, you can say, limitations. So mechanism of pharmacoresistance is another area where we can work and we can understand how the pharmacoresistance is there. We know different mechanisms have been reported, but still we are not able to fully understand why there is resistance to a particular drug. Then another area of this sub area is develop, refine, fully characterize and deploy epilepsy and seizure models that align with etiologies. So as I told you, most of the animal models, they may not be having good translatability because of their poor validity, either face validity, construct validity, or predictive validity. So we have to move further with the development of these models for better validity. Then identify, develop, and improve pharmacological, surgical, genetic, epigenetic, neuromodulatory, dietary interventions, and device to detect, predict, prevent, or terminate seizures or other epilepsy-related health risks while minimizing the adverse effects. So if you see all this, here we are talking about two things. One is the core epilepsy treatment is there, but is it taking care of the other comorbidities or the pharmacoresistance which is there? So that has to be given a priority. Then the last one, area 4, as per the benchmarks, is to limit, treat, or prevent co-occurring conditions associated with epilepsy across the lifespan in general and special epilepsy populations. Okay, so this is another interesting area because this is a rather orphan area in the case of epilepsy research. Many people are not working for these populations. Like understand and limit the impact of epilepsy on non-seizure outcomes such as neurodevelopment, mental health, cognition, health-related quality of life, and other functions. Most of the pharmaceutical industry is interested in getting the new molecules which are anti-seizure, and they are marketing them without taking care or without, you can say, taking into account what is the quality, improvement in the quality of life of the patients who are taking these molecules. So understand and limit the impact of anti-seizure treatments on non-seizure outcomes. Okay, then understand mechanism involved in non-epileptic seizures, and develop effective pediatric and adult treatments and assess outcomes of in non-epileptic seizures, including psychopathology and quality of life. So here we are talking about those areas, those gray areas where the research is minimum. Like SODAP is another area. Then impact of epilepsy on women health outcomes. Catamenial epilepsy is itself a big issue with the women because this is a sort of refractory, drug refractory epilepsy. It is not able, we are not able to treat with the available drugs. So this is another interesting area where benchmarks are directing us to do research. So understanding role of sleep, circadian rhythms, and in cognitive and psychiatric and other health related outcomes related to the epilepsy. So if you see all these areas, they guide us where we should pinpoint to do research. What was happening when we started doing research in epilepsy, what we were doing, we were just taking some molecules either from plant or from chemistry lab, and we were just testing on 3 models, rather 2 models at the time, PTZ-induced seizures and electroconvulsions, ECT convulsions. So these models were just telling us whether they're anticonvulsant or not. Okay, so if they are anticonvulsant, having anticonvulsant effect in ECT-induced seizures, then we were saying that it is similar to that of phenytoin type of drugs. And if they are effective in the PTZ-induced seizures, then we were saying they are more of GABAergic and they are useful for petit mal epilepsy. But these things slowly change. The research papers you see are improved now. These type of research papers are not, you see, available in the literature nowadays. They are trying to do more detailed study of. And let me tell one more thing to the students, that if we are going for acute studies on PTZ and ECT, definitely you can identify some molecules which are anticonvulsant, but you cannot say they are anti-epileptic. Okay, because epileptogenesis has not taken place in those acute models. For that, you have to go for chronic kidney models, where the epileptogenesis takes place slowly, and the animals are uh, fully convulsant or fully, you can say, epileptic, and that phenomena has to be there. So now coming to what we have contributed to these areas, a little bit that we have done, we'll be sharing. So neuropsychopharmacological investigations to reveal pathobiology of depression in epilepsy has been carried out by us. We really worked on comorbidities of epilepsy, psychiatric comorbidities. Depression was one, then cognitive comorbidity, cognitive deficit, which is associated with epilepsy. Then we try to understand what is the neurochemical basis of epileptogenesis. And then as the new trend is going on, we also try to use bioinformatic tools to understand what are the different drug targets through the lens of bioinformatics. And then we also try to develop an animal model for drug-resistant epilepsy using these bioinformatic tools. So let's start with the first thing first. We are working on two animal models. One is, if you see, chemical induced seizures, and other one is electrically induced seizures. In the chemical induced seizures, what we are doing, we are giving pentylene tetrazole, and at the dose of 65 to 75 milligrams, depending on the weight of this. And you see, the animal is, you can say, exhibiting and the seizures in this. And the other one, we are using uh, corneal kindling through electrically induced seizures. So here in this case, you will see the experimenter is using corneal electrode and inducing the seizures. And in this case, you see, after the application of current, you see what is the status of the animal. It is showing clonic, tonic, as well as rearing and stop tail. And you see, all these things are available in this. Is the clonic seizure, as you see. So these things are really, you can say, validated in the lab again and again before we proceed for any further investigations. So working with kindling, we notice that post-kindled animals are suitable to study psychiatric comorbidities, both cognitive decline and depression. Earlier, we found some papers where the people were using PTZ kindling model, whereby they were giving the drugs along with the during the kindling process. If you see, kindling process takes about 17 to 18 injections of pentylene tetrazole of 35 milligram per kg on alternate day at a gap of, you can say, 48 hours. And those kindled animals were treated along with the drugs which were not, which were supposed to protect the memory deficit and other things. But what we found that clinically this is not the same situation. In clinic, first we find that the patient are having epilepsy, and then after the epilepsy they are taking certain drugs, anti-epileptic drugs. Once they're taking anti-epileptic drugs, still they are having the problem. But the problem is either they are having depression or they are having cognitive decline. Or even if the epilepsy is left untreated, there are chances of cognitive decline and depression. So we are the pioneer in this, that we started using post-kindled animals for this purpose. So cognitive decline as comorbidity, if you see, recurrent disabling seizures reduce quality and expectancy of patient's life. This is clinical situation. Cognitive decline is most common psychiatric comorbidity in 40% of cases. And it has been reported that the lot of school dropout in the epileptic children. Anti-seizure drugs worsen cognitive. further worsen cognitive decline. You see, phenytoin is one of the drugs which is most commonly used drug as anti-epileptic drug, but it is affecting the, what we say, memory a lot. Then available cognitive enhancers are limited by seizurogenic potential. So if would like to add some seizurogenic, what we say, amniotic agents or nootropics, those nootropics probably they will be acting through cholinergic upliftment, and they are leading to reduce seizure threshold. So that is why they are seizurogenic. So that is not a viable, you can say, option to treat memory deficit in epilepsy with these type of drugs. So the magnitude of cognition deficit associated with epilepsy have been well studied. However, research efforts to find the remedial measure, you will find a lot of literature that pediatric patients, adults, even geriatrics, they are suffering from memory loss because of prolonged epileptic suffering. But the research efforts were very, very less when we started with it. So what we decided that we should first model that there is really a good cognitive deficit in the animal model. Whatever I'll be talking on preclinical research. So we consider evident deficiency of safe, effective management of memory deficit in epilepsy. So that's why we focused on psychoneuropharmacological investigations to reveal new pathobiology of cognitive deficit in epilepsy. For that, we first tried to see a model where we can validate the time and age dependent existence of cognitive deficit in chronic animal model of epilepsy, resembling to clinical epilepsy. And then to investigate neurochemical mechanisms underlying the induction of cognitive deficit in epilepsy by monitoring biochemical changes in brain of epileptic animals in above validated models. And then to explore the modulation of, you can say, different neurochemical outcomes, interactions to reveal common target for comprehensive treatment of epilepsy and restoration of cognition. This animation will talk about what we did. If you see, first part is a normal animal, whereby we had all the, you can say, level of catecholamine, monoamines, and amino acids at the base level. Even nitric oxide level is also at the baseline. So when we had epilepsy, this is the shift. The 5-HT, norepinephrine, dopamine, and GABA, they are reduced, glutamate is increased, and the acetylcholine level are also less, and nitric oxide levels increase. So when we give AEDs, this happens. Okay, so GABA is up, glutamate is down, acetylcholine is down, nitric oxide is down, and that leads to cognitive deficit. So now this cognitive deficit, when we try to treat with cognitive enhancers, what they will do, they will increase the acetylcholine levels, but at the same time, they will be provoking seizures. Seizure threshold will be reduced. So these are not safe for the therapy. But these appear like 5-HT, norepinephrine, DA, dopamine. They were supposed, GABA is already in the practice. So these three monoamines, if we can interplay with them, I think we can have the promising targets for comprehensive management of epilepsy and memory deficit. So what are the salient findings of these experiments? There is existence of age-dependent effect on learning and memory deficit in epilepsy. The chronicity of epilepsy is also one factor where if there's more chronic, then they'll be leading to more memory deficit. Recurrent seizures have more harsh effect on memory and associated neurochemical changes. That means the chronicity is important. And sodium valproate appears more suitable for effective management of epilepsy and associated memory deficit. However, valproate is associated with other severe side effects. So we were not considering it as an ideal drug. So phenytoin can be used along with tacrine for the management of memory complication induced by epilepsy or by venedone itself. But again, the seizure threshold will be altered. Then 5-HT2A, 5-HT2C receptor agonist and 5-HT3 receptor antagonist can be used for the comprehensive management of cognitive deficit in epilepsy. So these were the very good molecules for this purpose. So these are the publications which came out of this exercise. The first one where we have tried to, you can say, validate age-dependent learning in memory deficit in pediatric kindled mice. And then we have tried to see what are the neurochemical interplay in the brain, different areas of brain, the amygdala, hippocampus, cerebral cortex, for understanding what are the neurochemical changes which are really related to cognitive deficit. Then we have studied effect of different, you can say, interventions, like curcumin was one, and then saponin fraction from Vicus religiosa, and that was another study. And then we have also seen the adjuvant anticholinesterase therapy for the management of epilepsy-induced memory deficit. So there are some more papers which I could not include in this, but the idea is that this idea of understanding the cognitive deficit in epilepsy give us a very good research avenue. So similarly, the other researchers can also take a lead in this. Then coming to the another comorbidity, depression as comorbidity. I'm sorry. Majority of the anti-epileptic drugs or anti-seizure drugs worsen the depression. And depression is observed in the epilepsy. There was a report when one lady was having epilepsy and he was taking anti-epileptic drugs. When she was taking the anti-epileptic drugs for long, for example, 1 to 2 months, she started feeling depressed. What she was doing, she came to, because the patient becomes doctor to himself. What she was doing, she stopped taking those drugs, for example, 2 to 3 days. And then she has a little sort of seizure episode and her depression was over. So this is a documented, you can say, statement. So that means the depression was induced not only by epilepsy, but also with the prolonged use of anti-seizure drugs. So in contrast, antidepressants lower seizure threshold, as there was in case of nootropics. So antidepressants are also having a tendency to decrease the seizure threshold. So SSRIs have also been found to be ineffective in elevating depression in pilocarpine and carnic acid-induced temporal lobe epilepsy. SSRIs, you would say selective serotonin receptor reuptake inhibitors, have also been reported to accelerate kindling epileptogenesis. So these reports told us that we cannot use SSRIs as such, you can say, to treat depression and epilepsy. However, the mechanism of depression and epilepsy in the patients were poorly understood at that time. So neuropsychological investigations were carried out, whereby we had seen that what are the neurochemical and biochemical alteration associated with epilepsy and associated depression in cortical and hippocampal. These are the two areas which are most involved in the depression. Limbic brain is there. So to explore pharmacological modulations of underlying biochemical, there was another exercise which we have done. So if you see again, these are the two animals. One is kindled animals. K indicates kindled animals and V indicates the valproate treated animal. This is what happens in the case of kindled animal in comparison to NAV you're talking about. This is the neurochemical interplay. These things are increasing, these are decreasing. Kin is kynurenine, which is a metabolite from tryptophan through IDO pathway. And IDO levels are increased, nitric oxide levels are increased, and corticosteroid, they are also increased. And we see as stress is related to corticosteroid release and depression is because of that. So cortisone levels were also monitored in that. Whereas glutamate, GABA, norepinephrine, dopamine, 5-HT, and tryptophan, their levels were decreased. This was happening in the case of kindled animals. But in case of when we give the valproate to these kindled animals, this is what happened. The IDO activity was increased. That means the metabolic shift of tryptophan from serotonin to quinoline was more in that case, and that is ultimately giving us quinolinic acid, and that is involved in the you can say seizure induction. So if you see these studies told us that PTZ kindled animals were significantly bound to be associated with the depression. Uh, Elevated IDO activity as well as elevated nitrosative stress in cortical and hippocampal areas of the mice identified as possible targets, pathological reasons for association of depression in epilepsy. So chronic valproic treatment, valproic acid or valproate treatment in PTZ kindled animal did not show any significant restorative effect on elevated IDO activity and nitrosative levels in cortical and hippocampal area of the mice brain. Supplementation of minocycline, that is a IDO inhibitor. Okay, indolamine 2,3 dioxide inhibitor is with valproic acid has been found to safely ameliorate depression associated with epilepsy. Supplementation of selective 7-nitroindazole, which is a neuronal NOS inhibitor with valproic acid, have also been found to safely ameliorate depression associated with epilepsy. So based on this, we suggested that IDO pathway or the IDO inhibitors and NOS inhibitors may be useful add-on therapies to prevent comorbid depression in the epilepsy. So these are some of the publications which came out from this work. So this area is also explored in the right sense, where we can now talk about how we can control memory with epilepsy, depression with epilepsy. Then coming to working with these next thing which we have noticed, the keen observation. This is again a message to the young researchers. Whenever you are doing research, you should have a very keen observation. Whatever is happening, you should not ignore it. For example, when we were doing kindling experiments, what we noticed that when we start about 30 animals with kindling procedure, we were having a few animals left without kindling. They were not kindled. Okay, so this we say that there was natural kindling resistance in these animals, and most of the people do not even report it. But if we have the right observation and right recording, we can find out some new things from the research we are carrying out. So this natural kindling resistance give us an idea, like when we started with 100 mice, for example, 85% was kindling prone, good candidate for drug screening, because our aim was to screen new drugs or new molecules or to find neurobiology of the disease. So we wanted the animals to be kindled. And the 15% animals remained you can say kindling resistant. They were very poor candidate for screening limitation. My students used to say that, sir, 15% animals are wasted when we do it. Okay, so our effort is wasted in that. So then I said, no, we should use this 15% to give a better information for the future research. So what we did, in this line, we had the hypothesis that natural acquired neurochemical alteration in kindling resistant mice may work against epilepsy and might be beneficial. So new use of kindling resistant animal was carried out. We compared the neurochemical changes between the kindling prone versus kindling resistant animal in reference to baseline naive mice. So this was carried out through the HPLC with the ECG detector. So the neurochemical shift in naive, kindled, and resistant mice. You see, this looks to be interesting. These colors show excitatory, inhibitory, and modulatory neurotransmitters. N denotes the naive, and K denotes the kindled animal, and R denotes the resistance animal. And in the below, you will see the abbreviations for all which are used for different neurotransmitters. This is what happened in the kindled animal. You see, the inhibitory neurotransmitters were reduced and excitatory were increased, and the modulatory were also slightly decreased. Okay, what happens in the resistant animals? This looks to be more interesting. You see, this is what is happening in the resistant animals. In this case, you see, what is the difference? The GABA and norepinephrine levels were not reduced. Glutamine was not raised much. Quinolinic acid, which is end product of tryptophan metabolism through IDO, and that was decreased in this case. It was increased in the case of Kindred. And alanine levels were down, whereas taurine levels were up. Arginine taurine levels were up. They were down in case of kindred animals. So taurine modification was carried out in the subsequent study to prove our point that what we have found is good or bad. So we tested the anti-seizure effect of taurine per se and as a supplement with number of anti-seizure drugs with varied mechanism of action. And we used here, by the time there was a model available, lamotrigine resistant model of lamotrigine resistant model of pharmacoresistant epilepsy. We use 3 doses of taurine, 1500 and 200. As we know, usually in pharmacology, we are having triple dose regimen to make a, you can say, dose response curve. So taurine supplementation to anti-seizure drugs might be beneficial to overcome pharmacoresistance in epilepsy. This was the outcome. We supplemented taurine with lamotrigine, carbamazepine, phenytoin, and levetiracetam. You see, these are the results. Taurine restored the anti-seizure action of most of the drugs, which were resistant in case of lamotrigine resistant epilepsy. But it was ineffective per se, but when it was combined with the anti-seizure drugs, it was effective. See, so this shows that your keen observation may lead to a better, you can say, outcome in the research. So this paper was also published by us. Then our aim was, because there's not good work done in the area of drug-resistant epilepsy, so it requires ideal management, seizure freedom without harmful effects, early, simple, acceptable. Okay, so this is what the model should be in that case. So why to control seizures? Medications with or without surgery, nerve stimulation, etc. Sorry, ways to control seizures. We use medications with or without surgery and nerve stimulations. Use of medication is simplest. What we found, taking a pill is good than going for surgical interventions or electric, you can say, interventions. So it is the simplest, affordable, widely acceptable worldwide by patient and physicians. Problem and limitation with the use of medication is, as we told, drug-drug interaction, adverse effects, side effects, cutting-edge drugs are relatively safer. Limitations, these drugs are ineffective in 30% cases of epilepsy. Uncontrolled seizures, severe irreversible disabilities, premature death in drug-resistant epilepsy that we say SUDAP is there. So that means why we are having this, because we thought we are not able to understand mechanisms for drug-resistant epilepsy as such. That is one issue. And secondly, we do not have a good model for drug-resistant epilepsy so that we can test new molecules and we can have a better translatability of these molecules into drug-resistant epilepsy. So these are different mechanisms. Target hypothesis is there, the target is modified, for example. There's alteration in the drug targets, the protein structure. Then transport hypothesis. Here we say the reduced drug penetration or efflux of drug from the brain can be there. Then intrinsic severity hypothesis. Here the ASD resistance is secondary to intrinsic severity of excitatory and inhibitory balance and is associated with structural resistance. And then alteration in neuronal network, neuroinflammation, and blood-brain barrier dysfunctions. Okay, so out of that, what we can do, we can like here the change in this, we can't help it. We have to design the drugs for altered targets, then we can do it, and that is very difficult. So we can just specifically design one molecule for one type of patient and the other for other type of patient. Transporter hypothesis, we are talking about kinetic reasons, and kinetic reasons are not always the reason. So many studies have been carried out, but kinetic reasons were not able to resolve the issue. Intrinsic severity hypothesis is another one. Where this one has to be, so where we were, I think, And that's when this distance breaks. Okay, so as I told you, the different mechanisms of drug resistant, and we were thinking of how to make a drug resistant animal, and that was a challenge to us. So, whereas drug resistant epilepsy results from multiple mechanisms, some of them are unknown still and are difficult to fully acquire in the animal models. So, our objective was to transform the available genomic and proteomic information through system biology and network pharmacology approach into key determinants of drug resistance, useful for the development of an animal model of drug-resistant epilepsy with a good predictive validity. So this was the aim. For this, we went to search for the big data based on epilepsy, whether it's genomic data or what we say proteomic data, which is required to us. But this data, as we know, big data is easy to say we should work on big data and all those things. But this big data is having a lot of cacophony. This is called noise. And this noise, we cannot hear the right thing in the noise. So that was the issue. So because of that, listening to big data and making sense out of it became a challenge when the data transformed into bundle and stacks of unorganized, unstructured data sets. The converging data becomes loud and noisy, and with big data, you don't hear the voice, you hear the cacophony. This is the reason for cacophony. So we started very, you can say, enthusiastically that we'll collect all the things and we'll do something. But when we started it, we found that the number of papers is huge information available on us. Okay, I'll show you. Okay. But surprisingly, what we found that this big data is being tackled by bioinformatic tools, and that is boon for us. So this bioinformatics, what it is, basically collection of biomedical data, storage of biomedical data, organization of biomedical data, then retrieval of biomedical data on demand. Then you can manipulate this biomedical data as per your demand. Then modeling of biomedical data is there with the help of algorithms for visualization, analysis, and prediction using different algorithms and softwares. So we thought that if we can go this way, we can learn something based on this, and definitely we can use this data in a constructive way for our aim. So this is the scheme, what we did. I'll just, because I can't tell whole of the experiment, so I'm just trying to abstract it out. We did two things. First thing, we tried to find the genomic and proteomic data related to the epilepsy. This is one thing which is there in this part, orange part. Here we are talking about collecting the information about genes associated with epilepsy. And to augment this information, what we did, whatever are the available drugs, FDA-approved drugs, we try to find those drugs and make a database of those drugs and try to see how many drug targets they are interacting with. Okay, so and then we were making this common. 58 targets were common out of this. So 601 and 175 drug targets. When we totaled out, the protein associated with 543 out of 601 genes could be investigated for animal modeling in drug-resistant epilepsy. This was the outcome. And then we have used different tools. Diamond network approach is there, gene ontology. Then David is an open source feature where we can talk about gene enrichment, gene pathway enrichment, CAG pathways are there. So these are some open source tools which you can use, and we can churn the information in the desired form. And based on that, we have selected a disease target. You see, this looks very easy, but it takes time. We tried to select a disease target, and then we try to model that target in the animals to find truly resistant model to different anti-seizure drugs with varied mechanisms. Okay, this target was okay. So a little glimpse of what we did. This is a web service, DisGnet. If somebody of you know it, then it is better. Now, this DisGnet is nothing but a database and web service where you can find genes associated with different diseases. To see what happens. When we search it, we have to fill either the gene, SNP, single nucleotide polymorphism, or disease name. We put the disease name and we try to search. And then there was like this. You can have number of genes associated with that disease, and that you can This is, you see, the list which we got downloaded on that. And then we try to further augment this through drug central. We asked that epilepsy, how many drugs are available. So they give a lot of drugs. So from the drug central, we had a list of drugs. This list of drugs were put into drug bank database, whereby we can find what are the different targets for each drug. So here you see valproic acid we put, and valproic acid has got 7 targets, for example. So these targets were added to the genes associated with the epilepsy, and as I've shown that there was about 73 or 78 targets which were common, and those were subtracted, and we had 503 you can say genes which we can play for further analysis. So this is the IDs of different targets which are available for the FDA approved AEDs. If you see, this is the detailed analysis of targets. If you see, this is like 0, 1, 2, 3, 4, 5, 6. Okay, and these are the, if it is going up to this, that means it is having 6 targets. If it is going up to this is having 4 targets. So these are the drugs which are discovered chronologically, and their targets revealed till date are like this. Up to 2017, this data is. So these targets were chosen for studying further. And step 1 and step 2 we have done. In step 3, we try to see what is the interaction of these proteins, protein-protein interaction. So while we were doing this, we used INECT, and that is a very good web service, whereby we put list of all these genes which are presumed as proteins, because genes will be leading to the formation of proteins. So these proteins were then, they show, like if you see, 4, 8, 6, 6 interactions. So these interactions were important for us, because based on these interactions, we'll be analyzing further. We'll be visualizing these interactions, and then we'll be going for visualizing, we have to download this. So I have downloaded in the XGMML, you can say, mode of this particular information, and this can be useful for visualizing in the cytoscape. If you have heard of it, this is a network visualizing tool. There we can import and we can have the visualization of network. So it's this type of network you can see. And if you closely see, this is how different nodes are connected. And they are showing us what is the interconnection of these different proteins for the disease. And the next step was, once we have this protein-protein interaction network, then we have to analyze this. So this is one tool developed by Suzanne Gizian and their team. This is called disease module detection algorithm, derived from systematic analysis of connectivity pattern of disease proteins in the human interactome. This is like that they are trying to put whole of the interactome and then seeing our seed genes where they can interact. Seed genes are those genes which we have selected, like 503 genes, how they are going to behave in this interactome. So again, you can download this information, and then you can use in the Anaconda, you can have Python. In the Python, you have to write that code, and that code will be detecting in serial wise 1, 2, 3, 4. 1 is the closest one to your seed genes, and the number, as the number increases, it is far from your seed genes. The probability of involvement of that protein is less. Less the number, more is the probability that it will be involved. The beauty of this algorithm is that it is giving you new proteins which are not known yet for the involvement in the disease. So that was the advantage we have taken from this. And as told you, based on what we say, this information, we selected mitochondrial complex 1 as a common culprit for leading to drug resistance. So we tried to model that in the animal model. You see, this is the normal pentylene tetrazole kindling in the presence of rotenone for modeling drug-resistant epilepsy. Okay, so this is also the procedure. If you see, epileptogenesis takes about 35 days and drug testing from 37 to 61 days. What happens? We are going to test a panel of drugs which are available till date. Okay, so the next is lamotrigine kindling in the presence of rotenone. The first was simple pentylene tetrazole. Here it is lamotrigine kindling, because this is called as drug-resistant model of epilepsy, but still 2-3 drugs are still not resistant in this model. They are showing the anti-epileptic effect. So that's why it is not very good model. So we are trying to improve this model. So lamotrigine kindling in the presence of rotenone for modeling drug-resistant epilepsy was there. Here we found that all the drugs almost were showing resistance. In the rotenone lamotrigine kindling model, this was there. Then coming to corneal kindling. This is another model which we have tested along with the chemoconvulsant model. So if you see this one, this is also having, these are the prototype animals shown that how they were showing the seizures. In the corneal kindling, if you see, we were having rotenone supplementation, and then we have seen what is the effect of the drug. So rotenone adjuvant kindling was developed as a new model of drug-resistant epilepsy in our lab. So this rotenone kindling model shows resistance to lamotrigine, levetiracetam, and carbamazepine, zonisamide, gabapentin, pregabalin, phenytoin, topiramate, valproate, and there are 2 drug combinations too. And this is clinically required. Your experimental model should be resistant to all different mechanisms of the drugs plus their combinations too. So we have used both the things to make it truly resistant to the existing drugs. As a result, drug screening through this model may uncover broad spectrum anti-seizure drugs with novel anti-seizure mechanisms, which is a pressing issue to deal with the drug-resistant epilepsy. So this model seems, we think it, but this will be proved with time. Suitable for medium throughput drug screening, as it requires only 15 days. When we are talking about corneal kindling, it takes about 15 days for epileptogenesis and 1 day for each drug testing without any surgical procedure and social isolation of animals. And overall mortality rate is less than 10%. That is another advantage of this model. With the same approach as I talk about bioinformatic approach, we have also tried to find drug targets for the management of drug-resistant epilepsy. So this is another publication based on that, which was earlier we have carried out. So now coming to what, this is again, if you go to NIND's website, it is written there, what can I do? Okay, so what can you do? Log into NIND's crowdsourcing platform and see what researchers, clinicians, and people with epilepsy are saying about the priorities. Do you agree with the new benchmarks or not? You can comment. What is missing from them, again you can comment. You can also submit your own idea, give kudos to a posted idea or comment on a posted idea and choose your research area if you want to work in this area. And people with epilepsy can also email comments to curingepilepsy2020@ninds.nih.gov. So this site is very, very open to improve the life of patients with epilepsy. With this, I stopped talking, and lastly, I acknowledge all the funding agencies for providing the funds to carry out research. And in the top of the figure, you will find there are some of my collaborators. Dr. Jan Shastri is there. So this he is now director of NIS CSIR Institute in Johar. And Professor Vladimir is there right near to me in the photograph. He is my collaborator from Russia. Most of the bioinformatic work we have done together. And below we have some of my students who have contributed earnestly for whatever findings I'm sharing with you. One of them is in IPR Guwahati, Avnish Mishra, first one. Second one is in IHPT Palampur. He's a senior scientist there. And this Dinesh Gawande is in Creighton University as postdoc fellow. Priyanka is working in industry in New Delhi. Tanveer is again in Texas as a postdoc fellow. Sandeep is just completing his PhD. So thank you very much for your patience listening.
Thank you, sir. It was a fantastic session. and different disease you have touched upon. Mainly whenever we speak about the neurological diseases, everyone tends to speak about Alzheimer's, dementia, and Parkinsonism, which are quite nearer to each other. But listening about epilepsy and someone doing exhaustive research in this particular field was quite interesting to listen to. Thank you so much for joining us.
I
Over to you,
forgot to
Pranati.
mention that this is all blessing of my teacher, Professor S.K. Kulkarni. He is my
Yes, yes,
godfather.
yes. So, what better way to have a guru and shishya on the same platform?
Thank you.
That's fantastic.
I'm honored with this, man. So
Thank you, sir.
I am welcoming any questions if there are.
Yes, sir. I'm
Yeah.
taking up the questions for you. After a wonderful session, even I have a couple of questions. Let me take up the questions which have already been posted. The first question comes from Ms. Niharika Singh. She reads that when we shift the therapeutic approach from targeting seizures to targeting epileptogenesis, how can epigenetic modulations, especially mRNA modifications, be targeted?
You see, a lot of research is going on miRNAs now, which are involved in epilepsy. mRNA hypothesis is not, you can say, translating better nowadays because gene expression is better modulated by micro interfering RNAs rather than mRNAs as such. mRNA is simply a transcript from gene to, you can say, mRNA, and then that mRNA is having a lot of issues. It may be translated, it may not be translated. But what decides its translation is either siRNAs or miRNAs. So physiologically in human beings, we are having miRNAs. Those miRNAs, their level change. And because of that, that gene expression may be silent. So that may be the reason for, you can say, induction of disease. Most of the time, the miRNA panel for epilepsy is available. So for that, we need a whole new lecture where we can talk about miRNAs responsible for epilepsy induction and its prevention. mRNA itself, most of the people are doing study on mRNA. Gene expression means they are detecting mRNA levels. Okay, there are basically two levels you should detect if you are really interested in documenting that gene expression changes are there. So what one level is mRNA, second is the protein itself. If we are doing both the things, then it is confirmatory that gene expression is there. But most of the time, I've seen that people are talking about mRNA expression or mRNA, you can say, quantification, and that is not giving good results. The translatability is poor because those mRNAs may or may not be translated to a protein. I think I'm clear if.
I suppose she would have got her answer. She has just, she says, conveys, Niharika ma'am, she conveys thank you to you, sir, for the answer.
Thank you.
So I have a question. So in one of your researches, you have proven that sodium valproate stands as an effective treatment when we compare or when we have taken it into the kindled model. Sir, when we see the market survey, sodium valproate has several comorbidities also associated with it. I mean, even the dose of sodium valproate is very high. The first thing. Second thing, even dose dependent effect is also there with sodium valproate. Moreover, weight gain, sudden weight gain with sodium valproate. So, what is your take on this?
I already mentioned in my talk also that sodium valproate is having a number of side effects. That is why we cannot use it priorly for different things. Although it was effective in patients with the animals with memory deficit, it was not yielding much of the memory deficit. But we are avoiding its use because it is having a number of other side effects. I can't claim that whatever pathway I will be devising will not be having any side effect. You never know a molecule, what are its targets. You may be having only information about 1, 2, or 3 targets, not more than that. But nowadays, repurposing is drug repurposing is at its bloom. You can say you can have, you can listen to any drug like this drug can be useful in this drug can be useful in this. The reason being, we are revealing new targets for the existing molecule. This is another approach whereby we are trying to, you can say, fasten the drug discovery. Drug repurposing is there. So, like sodium valproate, when we started, it was treated to be, it was felt that we have got wonderful molecule to control seizures, right from in any age, from children to even pregnant women and then elderly. It was a wonderful molecule. But slowly, when we started using it, there was a problem. You see, statins. Statins are very good molecules, I think. In our era, if you see, statins were taken as a very wonderful molecules. And there was an article, if you see, it was in Indian pharmacology, the statin wonder of the world, a bubble to burst. That was the title of that article. So that was telling that when we are, in every science, there's a fashion. There's a free radical era, there's oxidative stress era, there's nowadays genomic era. And so every era we go with the movement, with the flow. Okay, but later on we come to know that this was not correct.
Right.
This approach was not correct. Now you see, every one of you who is having in different research areas, he has talked about oxidative stress, free radicals in all the areas of research. Despite
Right.
whether it is relevant or irrelevant, we used to do it. So that is where I am pondering on that we should decide on which area India has to work in. There has to be a national consortium on that. That these are the research areas where Indian researchers are supposed to work and will be funding them. Now, what happens? The funding is wasted, according to me. Some of people are getting funding and they are doing a very small experiment, then stop doing that experiment without any conclusion. If you are starting to doing something in research, then try to conclude it and present it to the public that this is what we have done. And then the criticism on that can make you further better research. That is important.
Right. So thank you so much. So one more question. I'm working with my MPharm student on epileptic models. Actually, I'm from pharmaceutics basically, and we are trying for nose to brain drug delivery using nanoparticles as formulations.
This is another area, nano drug discovery. I could understand nano till date, but it is really an emerging area.
So my question actually lies, we are working on the drug midazolam. Actually, this is recently new into the market, has a short half-life. And so the question is, the animal model which we have selected is, again, the PTZ-induced kindled animals. But when I did my MPharm and I had used the same model, I found like even the dose of 35 mg per kg even kills the animal.
Definitely. Do you see,
so
research,
how to like to do that
is not only science. Research is not only science, it is an art too. You see, you have to monitor closely when you are kindling that. We have to go up to the stage 4 or 5. Okay.
Right.
If you are going beyond that, then definitely you have to reduce the dose also. Like we are giving 35, it's not fixed that you have to give 35. You can start with 40, then you can come to 30, even 25 sometimes. In one day you can go for that. So what your aim is that there should be recurrent stage 4 to 5 seizures 3 to 4 times, so that you can prove that your animal is kindled. So for that you can do anything. You should not have a fixed chart that we have to follow that. That definitely mortality will be there. If we can observe that mortality is there, we have to first think of that how much, what is the stage, racing scale mein kitna uska aa raha hai. Accordingly, we can move on. And we should not, agar 5 aa gaya, dobara fir aap wahi dose denge, the chances that there will be mortality. So you should reduce it. And one more thing, I think no one will tell you what is a kindled animal. Kindled animal, people are saying, I have gone to certain meetings of animal ethics committee and people say that we are developing a kindling in 1 week, 10 days. So I'm asking them, this is impossible. I have spent about so many years in this area, but how can you prove that your animal is kindled? He said there were 2 times forced stage 4, stage 5. So this is not the kindled animal. When we kindle animal, you see, we keep them, we have kept maximum time period we have kept was 6 months. And after 6 months, again, we have given the same challenge dose, 35 milligram, and they were having fully blown seizures. If you see in the literature, they have written 2 months, they have written at least for 2 months, your animals should be in a kindled state after completing your kindling. Okay, so we have gone up to 6 months and we have seen that the things are working. So if you are going like jaldi hoga to jaldi khatam hoga, fastly get some effect, fastly it will be vanishing. So slowly we have to, because this is remodeling. What kindling is, this is basically remodeling of the brain, and remodeling you cannot have in one shot. For that you have to slowly change it. The more slowly is you're doing it, more better your animal model will.
Thank you so much, sir, for that insight. Because in most of the research papers I have gone through, read about inducing kindling in just 10 days, 11 days. Nobody has even talked about 2 months or 6 months period of time. I mean, the induction of.
No, basically, what is the purpose of doing kindling? We have to make it epileptogenic.
Yep.
I told you when we started the research, we were just doing. We were taking some molecules, some from chemistry labs, some from the plants, and we were testing PTZ-induced seizures, electroconvulsive seizures, the electroconvulsometer, and we were trying to publish paper that these are good anticonvulsant molecules. At that time, we were using interchangeably anticonvulsant and antiepileptic.
Right.
Now you see, as per the new classification, there are anti-seizure drugs. Or earlier we were saying anti-epileptic drugs. So this misnomer has been changed now. This is anti-seizure drugs. Because anti-epileptic drugs means your epileptogenic threshold will be reduced. You won't be having epilepsy, but we are not having any ideal anti-epileptic molecule to date. Okay, we are having anti-seizure drugs. So these things, when we evolve in an area, then we come to know. That is why it is important to hold an area and go through that for a long time, because you evolve in that. But nowadays, I have seen young generation, they say one paper in this area, the other paper is another area, and they are throwing papers like anything. So paper publishing is good, but it should not be the aim of your research, only aim of your research. So you have read my biodata. I have just 93 publications in so many years. So it hardly matters me how many papers I have. The only thing is I should be satisfied with what I'm doing. It is more important.
So thank you so much for that, answering my small queries and in a very depth way. And ma'am, do you want to add on something?
That punchline was something that is take home for, I perfectly agree with you. The number of publications, and this is a good message for all the young researchers, that quality of research and the quantum of research, and at the cost of what. So this balance one has to strike, and that's a very important message, sir, that you have given to the budding researchers, because tomorrow we are going to have our oral presentation session for the budding researchers of UG, PG and PhD. So apart from the scientific know-how of epilepsy, what you have just now said will be remembered by everyone. Thank you.
Thank you very much.
Thank you so much, sir. We would love to see you at Amity also whenever the workshop or conferences or seminars are conducted in a physical mode.
Definitely.
Thank you for your presence. And
no, Amity campus is now nearby us also. In Mohali, you are also having one Amity campus.
Yes. That's
we would like to have you at Lucknow campus, in fact.
Yeah, I have been to Lucknow many times. One of my students was in Naipur Library, which was shifted to Lucknow. Now he has moved to Naipur, Guwahati. He has also applied to you, ma'am, Avnish Mishra. I think if you remember.
Avnish, we had selected, sir. He has joined somewhere. Lovely mein hai wo abhi.
No. He is in Naipur, Guwahati.
Yeah, पहले lovely में थे, but his application selection हो गया था,
लेकिन join Amity. I said when you are going to Naipur, so why to shift too frequently? Go to Naipur.
Yeah. No, Naipur is a better place, of course. Yeah.
Thank you so much, sir. Thank you once again.
Welcome.
For your precious time. Dear participants, the feedback link has been posted in the chat section. I request you all to please fill the feedback form as soon as possible.
So may I leave the meeting, ma'am?
Thank you once again, sir. We look
Thank you.
forward to your visit to our campus in the future.
Sure, ma'am. Sure. Thank you.
Thank you
Thank
so
you,
much
sir.
for joining us. Pranati, you can make announcement about tomorrow.
Yes, ma'am. Yes, ma'am. I'm doing that only. We have two brilliant technical sessions coming up tomorrow from the speakers, Dr. Hassan Tahir, who is a TEDx speaker and neuroscientist, founder and CEO, California Institute of Behavioral Neurosciences and Psychology, USA. And second technical session tomorrow would be of Dr. Shubha Shukla, who is scientist, Division of Neuroscience and Aging Biology, CSIR, Lucknow. So we request the participants that you join us with the same enthusiasm as you did today and put in your questions, your doubts. And obviously these sessions will be followed by a post-lunch session of virtual oral presentation. Separate tracks have been made depending upon their specializations and the category of posters which we have, oral presentations which we have received. You would be directed in your WhatsApp group regarding the same. I say thank you to all of you and looking forward with a fresh start on the second day of the conference. Thank you once again, everyone. Do fill the feedback form, please.
I do want to add something to the presenters of tomorrow's session. In the post-lunch session, we will be having the oral presentation session, like told by Pranati. We will be having it in 4 different tracks on 4 different MS teams. So I request all the participants who have received the acceptance of their abstracts to be ready with their first thing. They have to email their presentation to the given email ID. On the WhatsApp group, you must have received certain very important instructions. I am going to repeat a very important instruction for all the presenters, that on the first slide of your PPT, please mention only the name of the presenter and your code which was there in the acceptance email. Please do not mention the names of the co-authors, names of your guide or mentor or your institute or your university. This is for unbiased evaluation of your oral presentation that we are taking this particular precaution. Please don't worry, your abstract has been already a part of the abstract book where the names of all contributors, the guide, the co-authors, your name of institute, corresponding author, everything is properly mentioned in the abstract book. This precaution we are taking only for the oral presentation session. Second instruction for you is, when you start your presentation, you will only introduce yourself as is the habit. You start telling from which institute you are, under whose guidance you have done the work. Please avoid both the things. You can practice today of not telling these things. And one most important, because we are having paucity of time tomorrow, and under each track we have not accepted more than 15 abstracts. The time is very, very important. Only 8 minutes to present and 2 minutes for question answer, each oral presentation of 10 minutes. If the presentation goes beyond 8 minutes, you will be stopped. Because we have, after the oral presentation session, we have the valedictory function. After the evaluation, we are going to announce the results, and all the winners and runners in each track, we are going to announce one winner and one runner from each track who will be getting cash prizes. So we have to maintain the authenticity, the genuineness, and the transparency in all our dealings, and that is why we have to abide by timing. And please do not mention about the name of co-authors or your institute or university. And please, if you do not share your PPT with the given email, you may not be able to present tomorrow. So this is my earnest request to please share your PPTs with our organizing team on the given email ID. All of them will be uploaded from our side, because everyone has got a different network issue, Wi-Fi strength. So we don't want the session to be delayed because of these problems or the technical glitches on your side. Please try to understand. So we are helping you out for the smooth run of the oral presentation session in the afternoon. And I wish you all the good luck and best wishes for tomorrow's oral presentation, all the young budding researchers. Thank you. That's all from my side. See you tomorrow, sharp at 10 o'clock.
Thank you, everyone. Thank you, ma'am. Thank you, everyone. Meet you tomorrow.
Thank you. Thank you, everyone.